Hi, this is your genetics AP Biology basic review that also includes the chi-square statistics problem. And so we're going to start with the statistics problem, and then we'll go through all the more basic problems at the end. So you can skip the second half if you really know what you're doing genetics-wise, but I'm going to put in a couple of cool points about like non-disjunction, bar body, exon activation, tissue mosaic pattern, some things like that that are covered in the more advanced concept. All right, this is the key for the genetics review and practice AP biology. We're going to skip page 1 and all the basic problems, and we're actually going to run right to the last problem, number 18, because this is fun. This is where statistics meets genetics. So I had to choose a letter for the eye color, and I chose E. And so big E is the dominant red eye, and little e is the recessive white eye. And remember, this is sex-linked, so there is nothing attached to the Y chromosome. So I'm going to do the Punnett square. I've got a heterozygous female crossed with a dominant red-eyed male. And so that's x to the big E, x to the little e, crossed with x to the big E, y. Remember, nothing on the y. It's sex-linked. All right, so I run my Punnett square, and I've got x to the big E, x to the little e, crossed with x to the big E, y. Take those haploid gametes and put them together into diploid offspring. And the first thing I notice is that both of my females are red-eyed. So the only white eye is down there in the bottom right corner, but let's take care of another red eye. There's a red-eyed male. And so now I just have my white-eyed male. And so if you're a white-eyed fruit fly in this cross, you're a male. So 50% are red-eyed female, 25% are red-eyed males, and 25% are white-eyed males, and 0%, by the way, are white-eyed female. So what we have in a Punnett square is a hypothesis. What we need to do now is compare the real-world data of our fruit fly farming to that hypothesis. So a chi-square statistic is going to look at the importance of the difference between observed, our data, and expected, our hypothesis. The first thing we need to do is figure out something called degrees of freedom, and that's going to be used to look up our statistical values on a table. So degrees of freedom is the number of categories minus 1. So I have red-eyed females, red-eyed males, white-eyed females, which don't actually exist, and white-eyed males. So that's 4 categories minus 1 equals 3 degrees of freedom. Now I'm being cautious in including those white-eyed females that don't exist because technically they're still a category. So when I look at my actual observed data, let's start with my big population. My 50% are supposed to be red-eyed females. That's 43. Then I have 19 red-eyed males. There's my 0 white-eyed females, which is expected, and my white-eyed males, 21. And now I got to come up with my expected values of my hypothesis. And that is going to be percents of total. So I know that I'm supposed to have 50% that are going to be red-eyed females, and I know I'm supposed to have 25% that are going to be red-eyed males. I know I'm supposed to have 0% that are white-eyed females, and 25% that are white-eyed males. But how do I actually get the numbers? Not just the percentages, but the numbers. Well, I need to look over at my data observed, and I need to calculate the total. And if I add them all up, I get 83 fruit flies. So I'm just going to apply the 0.5, the 0.25, the 0, and the 0.25 to the number 83 fruit flies. I get 41.5 that are red-eyed females. I get 20.75 that are red-eyed males, 20.75 that are white-eyed males, and 0 that are white-eyed females. The chi-square statistic is basically, it's a mathematical summation of observed minus expected squared over expected. It's actually pretty easy. If chi-square is less than my critical value, which will be looked up on a table using my degrees of freedom, then observed equals expected. But if my chi-square proves to be larger than my critical value, well, then I have a critical situation. Observed is not equal to expected. So basically, by having this O equals E or O does not equal E, it turns the whole world into a binary, a coin flip. It's either heads or tails. So, it either is or it is not. And so that makes the world a little easier to understand in that context. And that's the beauty of the chi-square. Turns the world into a binary. All right, so I have to look up my critical values, and statisticians have put together all these probability values, and they have these huge probability tables. What I'm looking for is the probability at 95% confidence, probability 0.95. And I can look and I can find these numbers here for 1 degree of freedom, 2 degrees of freedom, 3 degrees of freedom. So I have 3.84, 5.99, 7.8, etc. And I'm going to make a quick critical values of the chi-square distribution table. And the value I'm going to use is that for 3 degrees of freedom, and that ends up being 7.82. So if chi-square is less than 7.82, then O equals E, observed equals expected. And if my chi-square value ends up being greater than 7.82, that means observed is not equal to expected. So I'm going to run my problem basically by looking at observed, expected, then O minus E squared, and O minus E squared divided by E. You could actually just run it as an equation, but I like doing it this way because you can visualize the flow, the workflow, the way your problem answer evolves. You notice I've got sigma down there on the bottom. That means when I'm all done with this process, I've got to add them all up. Okay, so O is observed. So I've got my 43 flies, 19 flies, 0 wide-eyed females, and 21 wide-eyed males. And E is expected, and that's the value based on my hypothesis, which is my Punnett square. So 50%, 25%, 0%, and 25%. So I just got to grab those values from my worksheet, and that gave me 41.5, 20.75, 0, and 20.75 that would be expected. I know 0.5 and 0.75 fruit flies aren't that pretty to look at, headless fruit flies, but we still use those numbers, I think. We could round 42, 21, 21, but we end up with more than 83 flies. So I'm just going to subtract expected from observed now. And in the first case, I get a positive number, 1.5. The next one, I'm going to get a negative number. And that's not a big deal because when I square it, it'll go positive again. Negative 1.75, then 0, 0, 0. And now with my wide-eyed male fruit flies, that's going to be 0.25. Now we're going to take O minus E squared. We got to square all our values. And now I have to take those values from my squares and divide them by expected. Whoops, made a mistake. So an order of magnitude, 0.05, 0.15, 0, and 0.003. Those are very, very, very small numbers, which right away tells me, hey, look at this. If I add all these up, I'm still less than 1. So I'm not even close to 7.82. Well, let's add them up just to be good sports. And so I'm going to take my 0.05 plus 0.15 plus 0.003, and I get 0.203. So 0.203 is a really small number, and that's a lot smaller than 7.82, which is my critical value. So 0.203 is less than 7.82 from my table at 3 degrees of freedom. Here's the information so you can look at that. See, I'm not cheating. There it is. 7.82 was my critical value. I'm way under that. So this really helps verify the hypothesis that the eye color is sex-linked and that these fruit flies are demonstrating a dominant sex-linked trait. So here's the comparison again. Chi is 0.20, critical 7.82. Chi is less than 7.82, a lot less. And so my hypothesis was that if chi is less than 7.82, then O equals E, observed equals expected. There is no significant difference between the theoretical, hypothetical population of the Punnett square and the actual individuals that existed in the lab. Observed equals expected. So remember that my critical value was 7.82. That's for 3 degrees of freedom at a probability of 0.95 or 95%. What that means then, that remaining 5% could be by chance. So there's only a 5% chance that this is by random chance, that this thing I'm seeing isn't a real thing. And so there's a 95% chance that this supports eye color is sex-linked. Okay, we're going to look at something now called a choice chamber. And those are used in animal behavior studies. A good candidate would be like a little sow bug or pill bug that you might find in the basement or under a log somewhere, your little roly-poly friend, or a little crustacean. Now, when you do a choice chamber, things are a little different. You're seeing if the animal has a significant choice, and you're going to place them in random groups, basically evenly numbered groups on both sides, and find out if they sort of redistribute themselves. So when O equals E, that means they didn't care. And when O is not equal to E, that means they had a preference. Your hypothesis in this case is they don't care. And you're going to start off with, let's say we have 10 pill bugs because that's our minimum biological sample size. And I put 5 on one side and 5 on the other. If O equals E at the end of things, if you had 5 and 5, they didn't care. But if O does not equal E, now that means that they decided they wanted to go somewhere they like better. So I can make one side something they might like better. Maybe I'll put in some woody debris. It's holding some water. It's got some moss in it, some good food, some good smells. And so do pill bugs prefer to be in damp, woody debris with all kinds of food and smells? If at the end of this run, now I have 10 over there and 0 on the other side, then there's some data. I could also enhance the deal by covering it up with with a dark cover, or I could confuse them by seeing damp woody debris on one side and dark plain old plexiglass on the other, and see what do they prefer more. Do they prefer dark or damp woody debris? So I can also make a 3-chambered choice chamber, and that helps me to sort of focus on the different variables as being different from just plain plexiglass. So I can have the woody debris on one side, I could have the the woody debris on one side, and I could have the black screen on the other, and I could have all 10 in the middle in some plain old plexiglass. And then I can see, well, where do most of them go? I could run a hypothesis that basically says it's going to be 33%, 33%, 33%. Now with the first one, where I'm just looking at 2 choices, I have 1 degree of freedom. With the second one, where I'm looking at 3 choices, 3 minus 1 gives me 2 degrees of freedom. So you can see that a statistician begins thinking about the statistics during the design phase of an experiment. And that's going to be really important in science as you move forward. I'm also thinking about, I have a small sample size, so I need to use that 95% confidence, p equals 0.95. And so I look up on my degrees of freedom, my critical value table, and I get a critical value for my first experiment of 3.84 and 5.99 for my second experiment. Again, that's from the stats table based at p equals 0.95 at 1 degree of freedom and then 2 degrees of freedom for my different experiments. So then once I collect the data, I can run that data through and I can think about, hey, what do they prefer, debris or dark? Or debris or plain? All right, so let's move on now and look at our general practice. And we're going to start with multiple choice. And that first one, remember the keyword is hetero. D, heterozygous, means both kinds. The next one, a cell contains only half of the genes. That's haploid. Possessing 2 of the same gene, personal attached earlobes. 2 of the same is homozygous. Homo equals same. Number 4, a Punnett square that contains 2 different traits and measures 4 by 4 is a dihybrid cross. Di equals 2. Variety of a particular gene, usually half the information, is an allele. Highly organized, tightly wound strand of DNA is a chromosome. The genes that are attached to the X chromosome are sex-linked. The cell containing the full complement of DNA, both sets, is diploid. Again, di in diploid equals 2. The next one, a Punnett square that has just one type, is a monohybrid cross. And last, a trait that's always expressed when present is dominant. DACFIJHBEG. The next problem, number 11, is actually really easy. For any dihybrid cross that's heterozygous, remember you have 9 out of 16 that are dominant-dominant, 3 out of 16 that are dominant-recessive, 3 out of 16 that are recessive-dominant, and 1 out of 16 that are recessive-recessive. So all you had to do is look at what the question was asking and choose the one that fits. Is it dominant-dominant, dominant-recessive, recessive-dominant, or recessive-recessive? All right, this Tay-Sachs disease problem really just spells out your absolute basic monohybrid cross that's heterozygous. Big T, little t cross the big T, little t. The next one is color blindness, and color blindness is much like our fruit fly problem, where it's attached to the X chromosome. In this case, X to the E, X to the little e is a female who's a carrier for color blindness, and she's paired with a male who is normal visioned. But remember that Y chromosome doesn't carry the gene. So when the two get together, we're going to have X to the big E, X to the little e, cross with X to the big E, Y. The Punnett square is going to give us one female that's X to the big E, X to the big E. We're going to get one female that X to the big E, X to the little e. She's a carrier. We're going to get one normal visioned male, X to the big E, Y, and one colorblind male. So the only one who's actually colorblind in the end here is a male. And that's why males are so much more often colorblind than females. All right, this next problem is on the rule of multiplication. And the rule of multiplication considers the probability of events that are combined. If you flip a coin, heads or tails, it's 50-50. Let's say you get heads. What's the chance going to come up heads the next time? It's still 50-50. And if you flip it again, it's still 50-50. But what are the chances of getting 3 heads in a row? Well, that's 0.5 times 0.5 times 0.5. In the rule of multiplication, what I'm looking at doing is combining the probabilities of events. So in this case, what are the chances that I get a certain set of traits? To do this, I set up the simple monohybrid crosses for each trait. And so I set up my A's, and then I set up my B's, and I run my Punnett squares the way I'm supposed to. Let's set up my C's as well. You know, I really don't like C in a Punnett square because a little c and a big C, if you're sloppy at writing, it can really mess you up. So really make sure your little c's are little c's. Now we're going to move in, we're going to do our C's here, and when we finish this up, we're going to have all the possible outcomes for the A trait, the B trait, and the C trait. So now if I want to look at a particular set, I just look at the probabilities. Let's start with A here. We've got A is 50% dominant, B 50% expressing as dominant, but C is 100% expressing as dominant because we have homozygous dominant and heterozygous. So I've got a 0.5 times a 0.5 times 100%, which is a 1.0. So the probability of getting these 3 dominant traits together is going to be 0.5 times 0.5 times 1, which is 0.25. So there's a 25% chance that I'll have the earlobes, the tongue roller, and the widow's peak. All right, the next one is a really simple one. We're just looking at what is the outcome from crossing a pink snapdragon with a pink snapdragon. Remember, this is the rule of incomplete dominance, and all we're doing is we're crossing a pink, which is carrying an R and an R prime allele, or a red allele and a white allele, and we're crossing that with another pink. And we're going to get 25% RR, 50% RR prime, and 25% R prime R prime. That's a white snapdragon. That's such a basic problem. There it is. 25% are red, 50% are pink, and 25% are white. That's the snapdragon problem. Piece of cake. All right, this next problem is actually really interesting because it deals with something called Barr bodies X inactivation. And so when a sperm fertilizes an egg, and we're looking at a sex-linked trait, if the sperm is carrying the female chromosome, the X, and it fertilizes the egg, you're going to get a female. So you'll have XX. And if the sperm is carrying, in this case for black cat, the allele for black, and the egg is carrying O, you're going to have an XBXO. And so when that cell divides, each daughter cell is going to be carrying one activated X and one inactivated X that becomes something called a Barr body. And so you start to get clusters of cells that are going to express black and clusters of cells that are going to express orange. And so this creates what's called a tissue mosaic effect, where certain clusters of cells that are expressing black fur for the cat are going to express black, and certain cells that are expressing the orange fur for the cat are going to express orange, and you'll actually see the ancestral patches of cells from that original cluster of cells that turned into a cat. So that's called a tissue mosaic effect. Now the problem talks about an orange cat that breeds with a male black cat. And the male can either pass on an XB and make a daughter, and so we're going to have an XB, XO. Now mom can only make an O because she's only got XO or XO. So when the dad cat gives that XB to the XO, now we have a black and orange female cat. The dad can also pass on his Y chromosome and have a son. In this case, that Y chromosome doesn't carry anything. So then you get an orange male cat. So now we have two choices, a black and orange female or an orange male. But the problem talks about a black and orange male cat. Where does that come from? First thing we should do is prove this with a Punnett square. Just make sure we're not making any mistakes here. There's my black and orange female, and there's my black and orange female. There's my orange male and my orange male. I still don't have a black and orange male. So what's going on here? The answer is non-disjunction. And that's a process. To understand, I'm going to need to look at the process of meiosis in its purest form. And remember that meiosis is the construction of sex cells, haploid sex cells. And so I'm going to start with a diploid cell, and I'm going to go through what's known as a reduction division. You'll also notice that the ancestral chromosomes stick together in meiosis. So my X's stuck with my X's, my Y's stuck with my Y's. And what results is, the expected. But from this male, look what happened. Early on in that first cell division, the Y stuck to the X. On my other cell, for that chromosome, I've got an empty set. I've got nothing. So even though I have haploid cells and one cell has a bunch of chromosomes, it's missing all of the sex chromosomes. And the other cell is doubled down on sex chromosomes. So when I go through my final reduction division, I end up with the empty set over here. And then over here, I end up with an XBY and an XBY. The entirety of the male's sex chromosome are represented in the sperm. So in a sense, you've got a haploid sperm with all those sets of chromosomes have been reduction divided, and you only have half, except for the sex chromosomes. You've got a diploid set. So what happens when you have a diploid sperm that fertilizes a haploid egg? Well, 1 plus 1/2 equals 1.5, or 3/2. This is non-disjunction. Non-disjunction starts in the sex cell formation and ends up carrying on as a chromosomal abnormality in the fertilized egg. So here I have XO, XB, Y. So in this case, I have a black and orange cat. And it has a Y chromosome, and so it will express as a male. And that's actually how you get calico male cats, through a process known as non-disjunction. All right, let's look at blood type. Blood type is really unique because there is a recessive allele, that little I, that ends up not coding for any blood antigen, and then a codominant situation where you can either have the A antigen or the B antigen or both. So blood type then is for a protein recognition on the cell membrane, and it's a codominant trait. You can have A, B, or both. So let's look at the genotypes to phenotype transition here. Make a list. If you don't have any dominant alleles, you'll have type O blood, and you actually end up not expressing any antigens. If you have either of the combinations for the A antigen, you get A, and you're type A. And then if you have either of the B antigen combinations, either I to the B little I or I to the B, then you have the B antigens, and you're type B. If you get both, I to the A, I to the B, then you're blood type AB, and you have both antigens. All right, some people wonder, well, what's with the AB positive or, B negative or O positive, O negative? Well, that's a different protein, and that's a protein that we refer to as the Rh factor, and we're not considering that right now in this problem. All right, in our question then, we have a dad who's type A and a mom who's type B, and the question is, we got a kid who's type O. Can this work? Well, since I'm seeing two little I's, I have to choose the diploid combination from my parents that has a little I allele. And so if I run a quick Punnett square with my little I alleles, then I get four offspring, I to the A, I to the B, I to the BI, I to the AI, and a little I. So we have a type AB, a type B, a type A, and a type O potential in the kids. If dad is I to the A little I and mom is I to the B little I, they can get all four different types of blood types. So this tells us two things. First of all, dad and mom are heterozygous because they had a type O kid. And yes, dad is actually likely to be dad. Thank goodness. Congratulations, dad. All right, I hope you found that useful. So, hey, we'll see you.